Guys, we are going to be using molecular evidence to compare two species with each other. So what do I mean by this? The two molecules that we can use to compare living things and their evolutionary history are their DNA base pairs and their amino acid sequences. So we can look at the base pair sequence of a similar gene between different species and count how many ATCGs that they share in common. Or we can take a shared protein that is slightly different from species to species and take the amino acid sequence and compare those. So the key point will be the more similarities that two species share in their DNA sequence or amino acid sequence, the more related they are in evolution. So this lizard would have more DNA in common in terms of the ATCG sequence with this bird than this lizard would have compared to this shark lamprey down here. And the vice versa is true. More differences, the less related they are. So let's jump into an example here. Here we are looking at the living things that share the protein cytochrome C, which is a protein in your mitochondria that helps you make DNA. So I always want to figure out what is this data showing me. So I always go to my number one source, the title, cytochrome C evolution. And these numbers represent the amount of differences in the amino acid sequences. So I'm looking at differences. Sometimes it's similarities. You just need to know what these numbers are. And this is comparing it to humans. All right, so I have humans have 0 differences with a chimpanzee, so the humans are the most related to the chimpanzee compared to the other ones on this list. Humans are the least related to yeast cells that make bread because they have 42 differences in their amino acid sequence. And it just goes right here in descending order. All right, so if I were to make a cladogram like this, you don't have to do this. I'm just going to be using a scratch paper very frequently. You would make a diagonal line. You put your most advanced species right here, which is your human. And it is the most distantly related to the yeast. So I put the yeast on the opposite side. And it's the most closely related to the chimp. So I put the chimp right here. This would be a good visual way to show the data. I'm not going to write all the animals in here, but that amino acid sequence gave me the information I needed to create this cladogram. All right, now let's raise the bar a little bit. Okay, so I'm looking at 5 species of trees. And this is the outgroup. This is the one that we know is going to have the least amount in common with all the other ones, and it's going to be the animal on the far left, similar to where we had yeast on the cladogram last time. So I'm going to teach you now how to create a data table to store your numbers that you come up with. So I'm looking at species A, B, C, D, and we'll just label that outgroup. So I go to my scratch paper like this. Let's see if my writing helps this. I go A, B, C, D, and then I put right here out. I put that down the side, and then I put it across the top. A, B, C, D, out. And I like to have grid lines. Like that. All right, so no, I'm not going to fill out every single box on here because if you were to look at the final result, the top half, like let's say this is a sandwich and you cut it diagonal, the top half will have the same numbers as the bottom half. Let me show you. We're going to make a dividing line. You cannot compare something with itself. That's not a comparison. So comparing A to A does not make sense. So this is kind of like a Punnett square. Top and sides, bring them in. You can't compare something to itself. So these boxes are not applicable. So I'm putting an X through it. Now, this creates my dividing line. I'm either going to fill out everything above it or below it. I will get the exact same answers. In fact, I've even seen people do similarities in sequence up here and differences down here because sometimes you need the additional data. We are not going to do that. So I'm going to make this more visible by highlighting my dividing line, and then I'm going to begin counting. So this box right here, I'm going to fill in the number that shows, for me, I'm going to do similarities. I like similarities better than differences, but it's preferences. You would get the same cladogram. I'm going to look for similarities between A and B. So I'm going to go back to my chart and I'm going to count. This might be where it's helpful to print if you can. So let's see, I'm just going to count. Follow my cursor. 1 similarity, 2, 3, 4, 5. So here I record 5. Then I compare my A and my C. 1, 2, 3, 4, 5, 6, 7, 8, 9. Wow. So I'm going to put 9 right here. And then I want to compare A with D. 1, 2, 3, 4, 5, 6 similarities. And then I want to compare A with my out group. So I'm going to go, there's 1, 2, 3, 4 similarities with my out group. Okay, take a second, please, and pause me talking once I get to this slide, and I want you to fill out the remainder of these boxes with this data. Here is our answer. So what I've done is I've gone through my data and I've looked for large and small numbers. And my biggest number on here is 9. Species A and C have 7 similarities in their amino acids. So I know that on my cladogram down here, they should be close to each other. And my small numbers are 4, but there's many that have 4 similarities, so I can't really do anything with that number. So let's see. I'm going to make my cladogram. And I have 5 species that I'm comparing, so I did 5 lines. And I just write what I know. The outgroup is the least evolved and closer to the common ancestor, so I'm going to put the outgroup right here. And I'm going to ask myself, is there any conclusive data in my table to say which one is the most related to the outgroup? So I go to my outgroup column, and I look, there's the number 7. That's my biggest number. So it has 7 similarities with species B, so I'm going to put B as its next door neighbor. And these actually go in descending order, so I'm just going to use the order of the outgroup as my data to fill out this cladogram. So this one had 7 similarities, D had 6 similarities, C had 5, and A had the least similarities to the outgroup. So it makes sense that it's right next to it. And then it's also justified because A and C had 9 similarities and they are next door to each other on the cladogram. Another practice problem. So let's look at our species here. We're going to be comparing the human, horse, gorilla, chimp, and zebra. We will be using the sequence of amino acids in the same part of the hemoglobin molecule. So keep in mind, humans have a hemoglobin that is 15,000 amino acids long. So the horse, gorilla, chimp, and zebra might have a different nuance in their hemoglobin. So it's important that we take the sample from the same place in their hemoglobin molecule. So let's see, I'm going to create my data table. I got a new sheet of paper out. You do not have to. This is continuous notes. So I know that I have my human, and let's see, horse going down, gorilla, chimp, and zebra. So if you can see, I listed them down. Now I'm going to list them across the top. Okay, I'm going to make my grid lines. going to make my dividing line. Can't compare something to itself. That. And I've decided that I want to chart similarities in their amino acid sequence. So I'm just going to put similarities right there to remind me what I'm looking for. And I am ready to count. We'll do the first row together. So first comparison, human to horse. 1 similarity. Human to gorilla. 1, 2, 3. 3 similarities. Human to chimp. 1, 2, 3, 4. 4 similarities. Human to zebra. 1 similarity. All right, this line of data is not conclusive enough for me to make a cladogram yet, so I will need to fill out the other boxes. Please pause this video and fill out the remainder of the data table. All right, here's our data. So I'm going to make a cladogram with 5 slots. All right, and humans are always at the top of the evolutionary pile. So I'm going to put humans up here. And then I'm going to ask myself who is the most related to humans, so who has the most similarities. So I go to my human row. The chimp has 4 similarities, so I'll put the chimp next to the human. And is there a second runner-up? The gorilla, so I'll put the gorilla right here. All right, now I'm at a fork in the road. The horse and the zebra both have 1 similarity, so I need to ask myself a different question. I'm going to ask myself, what's the most related to the gorilla? The horse or the zebra? The one that's the most related I'll put here, and the least related here. So the gorilla to the zebra is 1, and the gorilla to the horse is 2. So 2 similarities, the horse is more related. So I'll put the horse here, and I'll put the zebra here. Okay, so what we're going to do right now, instead of counting amino acids, we are going to count differences this time in nucleotides. A nucleotide is A, T, C, or G between species A, B, C, D. Sorry, a lot of letters now that I think about it. So the cladogram that you are going to create is going to compare species D with the other 3 species. So whenever you are given an animal or whatever species it is to compare something to, that always goes at the top of your cladogram. So it would be like the lizard in this one. You'd put species D up here, and the relationships would follow underneath it. Okay, so let's see. Let's go ahead and do this very simple data table. Please be patient with my dot cam. It doesn't like to focus. So we do our species down the side and across the top. Throw in some quick grid lines. Cross out the ones that compare DNA with itself, and then title it, just so you remember, this is going to be differences in DNA sequence. Okay, so I am going to do the first one with you. I'm going to compare the differences between species A and species B, and then I'll have you fill in the rest on your own and answer on the Google form. So it's nice here that it's color-coded. So I'm comparing, if you see my cursor, species A to B. So I'm looking for differences. 1, 2 differences. So please fill out the remaining differences and answer on the Google form below.